I've been with the company about 6 years now. And what really excites me most and really why I'm still here after 6 years is really the technology, the fact that you can break some of the barriers that you see in a typical microbial system to deliver the kind of effective solutions that we've got, sustainable areas of great impact, is just really exciting for someone that spent their whole career in a bio-based industry. We've taken that technology and really focused on RNA. We started this journey in 2017. We built a plant in 2020, and we're going to get a product out on the market in 2022. I mean, for a young company to do all that and to have commercial product in a facility is just phenomenal. We have both the technology, the drive, the capability, and the expertise to get it done. So we've developed a platform based on RNA, and we're able to make many different RNA molecules. Many of the elements are common against all those different types of molecules that we're going to make. In a processing perspective, it allows us to actually look for advantages from one molecule type and apply it to all the others. And we're in fact leveraging back and forth, figure out how to make some breakthroughs in mRNA. We're able to actually take that back and make some tweaks in the dsRNA process, which is for an agricultural purpose. So the learnings, being in a common platform, are quite leverageable. So as we figured out how to build and scale in Rochester to make a double-stranded RNA for the agricultural markets, an industrial-based plant, there's still some critical learnings that we're going to be able to leverage as we're now considering how to design and build an mRNA plant for an injectable vaccine. Of course, the compliance and the regulatory are very different, but some of the basic processing elements, you do a fermentation, you do downstream recovery, are consistent against all the platforms. So the leverageability on the process is really to our advantage. To make RNA for the agricultural space, people have been looking at that for years, and they were unable to figure out really how to use a basic microbial system to produce a specific designed RNA. Just directly ferment it, and/or you use an IVT reaction where you feed nucleotide triphosphates. Both of those have disadvantages. One's got scale and cost, the other has a cost disadvantage. What we've been able to do is take it back a few steps where you can get really inexpensive substrates and actually energize those substrates and actually have them make your RNA of choice. That allows you to make metric ton quantities in a capital efficient system at a cost that the farmers can afford.